So after knowing the various steps involved in somatic cell fusion, that is, we know what is an explant and we know what is a nutrient medium in which we have both solid as well as liquid. Then with limited plant hormones like cytokinins and auxins, so we can going to get what is known as an undifferentiated cells. From these undifferentiated cells, we know how to generate a protoplast. All right. Now we will just discuss about in case of a recombinant DNA technology, where a specific gene of our choice is introduced. So we cannot do that in case of a callus, because callus do not contain any plasmid. All right. So a similar callus-like situation is observed in most of the angiosperms or gymnosperms, like gymnosperms are the non-flowering plants, and then we have the angiosperms, the flowering plants, in which the dicotyledonous angiosperms are going to be common with respect to this disease, what is known as a crown gall disease. So if you just take a callus, so now similar to callus, here you are going to generate callus in the laboratory in the nutrient medium. So we are going to have a similar undifferentiated mass on a living plant. So we call this particular one as a tumor. It is a uncontrolled cell division, but without any differentiation. A mass of cells are going to be generated. We call this as a plant tumor, and that can be defined as it's a lump of undifferentiated cells. Now, this particular plant tumors, it can be easily induced in both gymnosperms as well as in dicotyledon angiosperms using a microorganism. It's a gram-negative soil bacteria. Call this as an Agarobacterium tumefaciense. Okay, now this is a soil microorganism. It's a gram-negative bacteria. This can induce crown gall disease or a plant tumor in gymnosperms and angiosperms. So when you try to analyze this Agarobacterium tumefaciense, we are going to get two varieties. One, we call this as a virulence variety, or also oncogenic in nature. Another one is going to be the avirulence, a non-oncogenic. That means some will induce plant tumor and others will not induce plant tumor. So when they analyzed in detail, the virulence do harbor an extra chromosomal DNA, that is the plasmid. This particular plasmid, we call this as a TI plasmid, is basically because tumor inducible plasmid. This plasmid is temperature sensitive. That means it doesn't work optimally at 28 degrees centigrade, but efficiently it works at 37 degrees centigrade in fact. So now when it comes to this virulence and an avirulence, so we can take the virulence variety, and if you eliminate TI plasmid, the virulence is no longer capable of inducing tumor. On the other hand, suppose if you do have a virulence, to this virulence, if you harbor a TI plasmid, then virulence will become virulence. In other words, the virility or the capabilities of this Agarobacterium tumefaciense to induce plant tumor lies in the extra chromosomal, that is the TI plasmid. Okay. So now, in case of TI plasmid, what happens? How this crown gall, a lump of undifferentiated cells, is going to be developed? So now, we all know that in case of the plant hormones, cytokinins will transformed into shoot and auxin into root. Now, this particular TI plasmid, they do have virulent genes. So they make sure that this particular plant cell will not be differentiated into any form, either a shoot or a root or a bud or a flower. Okay, they want these cells to be undifferentiated and it is a lump of mass. So usually when a microorganism infect, we have a symbiotic arrangement. But here the beneficiary is only the Agrobacterium tumefaciensis but not the plant. Okay. Now this condition we call this as a parasitism. So in this, plant is not going to be the beneficial, but the microorganism is only the beneficiaries. We are also called this as a genetic colonization. This is because of this plasmid genetic. So we have the advantages to a microorganism but definitely not to a plant. Now when you try to analyze this particular TI plasmid, it is a circular plasmid, and this circular plasmid has got a size because they have isolated several plasmid from this Agrobacterium tumefaciensis. Now the size of these plasmids can vary from 140 to 235 kilobases, and all of them are all temperature sensitive. Now that is going to be the 37 degree centigrade. Works optimally but not at 28 degrees centigrade. Now, so once we do have this particular TI plasmid, this plasmid is present in the microorganism. Now this plasmid has to be transferred to a plant cell. I think we know that in case of a plant cell, so it has to enter through the cell wall and into the cell. Then it has to integrate it in the nucleus, because it has to enter into nucleus, and then this particular DNA has to be transformed into the plant chromosomal DNA. Now when it comes to the transfer, the entire plasmid is not going to be transferred to the plant chromosome. Only a part of the DNA is going to be transferred. Now that part of the DNA we call this as a tDNA or a transfer DNA. So we have to make sure that these plasmids harbors certain virulent genes. So because of these virulent genes, so the differentiation is going to be blocked. There is no way it can develop into a shoot, or there is no way it can develop into a root. So no, they are the virulent genes in fact. All these virulent genes are all present in the TI plasmid. Okay. Now we know that in case of this particular plasmid is in a prokaryotes. So now this has to get integrated into a eukaryote, and they have to express certain gene products so that this particular cell will never differentiate either into a shoot or into a root, so that it will just simply multiply as an undifferentiated cell mass. So for that, what happens, this TI plasmid should get integrated into the plant cell. During the integration, because the size is so big, not the entire plasmid DNA is not going to be transferred or integrated into the plant chromosome. Only a part of the DNA is going to be integrated. Now that part of the DNA we call this as a transfer DNA, or also call this as a tDNA. Okay, now this condition it's purely a parasitic conditions in which microorganisms are beneficiaries, and we also call this as a genetic colonization in fact. So we will try to understand this TI plasmid or the genetic colonizations. What are the characteristic features this TI plasmid does? First thing is going to be the tumor induction. Alright, I told you that this is parasitic in nature. So now it has to produce certain components which is useful for this microorganism to sustain. So that results in the biosynthesis of what we call this as opines. Opines are nothing but unusual amino acids. These unusual amino acids, these TI plasmid will produce the required enzymes in order to make these opines, because these unusual amino acids, they are the provider of nitrogen source and carbon source for this microorganism to grow. So they will not survive on the regular amino acids of the plants. They have to have these amino acids, what we call this as an opines or unusual amino acids, they are the nitrogen and carbon source for this microorganism to grow. So as a result, we have the catabolism of these opines. So all these three things are going to be induced by the TI plasmid. One is the tumor induction, so that it will make sure that it will not be differentiated. Second thing, for the survival of the microorganisms, they have to synthesize certain unusual amino acids, what we call this as an opines, because these unusual amino acids are the carbon source and nitrogen for these microorganisms. Then they need to have the catabolism of these opines. Okay, I told you this carbon and nitrogen source. What exactly are these opines? Because now the tDNA is also being well characterized, in fact. All right. So now, based on the opines, again I think we have different class of plasmids. Okay. So now one is octopine. So another one is going to be the naupolin. Okay. Now they are all the opines, which is unusual amino acids. So on detailed characterization, they found out that some of the TI plasmids will end up in the synthesis of an octopine, and others will end up in the synthesis of a naupoline. So both octopine and naupolines are the opines, which are unusual amino acids. What exactly happens? So in case of an octopine, now this TI plasmid has a very important enzyme called as an octopine synthase. Whereas in case of TI plasmids which produces naupoline, so they harbor an enzyme naupolin synthase. So octopine synthase and naupoline synthase are very, very important. So usually what happens, octopine synthase does is that so we have an arginine here. This is a regular amino acids. Now this arginine combines with a pyruvate, and that will create an octopine. Now usually what happens, like we have the alpha amino group. So now these alpha amino groups normally in case of a peptide bond fuses with a carboxyl group. Then we have the formation of a peptide bond. Whereas in case of the opine, this amino group fuses with this keto group. Now that results in the formation of an octopine. Now this is the arginine and now this is the pyruvate. Okay, so now these two things will end up in the synthesis of an opine. So now the keto group of pyruvate is important, the alpha amino group of an incoming amino acid is very important. That results in the synthesis of what is known as an octopine. So now the octopine produces several opines. I think I have just given you the other examples. For example, pyruvate with arginine will provide octopine. Simultaneously, this octopine synthase will also involve other amino acids. For example, pyruvate with lysine, that gives lysopine. Pyruvate with histidine, they are going to provide histopine. Pyruvate with ornithine, they are going to get octopinic acid. Alright, so now all these things I think is going to be the opines, octopines synthesized by the octopine synthase of the TI plasmid. So we also have one class of plasmid in which naupolins are going to be synthesized. So in case of a naupolin, instead of a pyruvate, the keto acid is going to be the alpha ketoglutarate. So now this ketone will involve a similar reaction with the arginine. So alpha ketoglutarate with arginine gives rise to naupolin. Okay. So now this naupolin synthase can also produce naupolinic acid with alpha ketoglutarate with ornithine. So these are the two major class of plasmids we have characterized, and basically because in case of the octopine, I think we have the octopine synthase. So now they preferably uses pyruvate with several amino acids to produce opines like octopine, lysopine, histopine, octopinic acid. Then in case of the naupolin synthase, which is another class of plasmid, they use alpha ketoglutarate with arginine produces naupolin, and with ornithine they produce naupolinic acid. These amino acids are not useful for plant metabolism, but these opines or these unusual amino acids are very critical for the survival of a microorganism. So what the microorganism does is that they will allow the cell to multiply so that they can provide you all these amino acids, so that the respective synthase, either octopine synthase or a napoleon synthase, utilizing these amino acids will convert them into the required amino acids. We call these unusual amino acids for the survival of the microorganism. So for the survival of the microorganisms, we do not want these cells to be differentiated. So we want these cells to be multiplied, because these cells will provide the required amino acid pool. Now using these amino acid pool, they are going to synthesize the opines. These opines are critical for the survival of microorganism, but it is not going to be useful for the plant. So as a result, so now this microorganism is parasitic in nature, and these particular plasmids do have the specific enzymes which will convert these amino acids into the respective opines, in fact. So based on this genetic colonization, TI plasmids, I think they have characterized quite well, and there are several kinds of plasmid. They produce different kind of opines, but the major opines which is required for the survival of microorganism is the octopine. Another one is going to be the napoleon. Alright, so now I think we will discuss about the genetic details of the TI plasmid. I told you these particular plasmids, I think they do have a size of 140 to 235, as I discussed earlier. Not the entire plasmids is not integrated into the plant chromosome. Only a small portion of DNA from the plasmid is what we call this as a tDNA. And we also discussed about the napoleon plasmid, and we also discussed about the octopine plasmid. The DNA which is going to be transferred, or the tDNA, the size normally is somewhere between 22 to 23 kilobases. So now then they have characterized this tDNA from napoleon as well as tDNA from the octopine. So in case of a napoleon, the tDNA transfer is just one segment. Alright, whereas in case of an octopine, so we do have two segments are going to be integrated. So one is a TL, that is the left segment. Another one is going to be the right segment. Now the left segment is approximately 13 kilobases, and the right segment is going to be the 7 kilobases. And the left segment is very critical for tumorigenesis or the virulence, but not the right segment, in fact. So now what happens, they have also analyzed how this tDNA is going to be integrated, because in case of the yeast DNA, we always talk about the homologous recombination. Now here, whether the transfer or the integration is also happens because of the homologous recombination or by a different mechanism is interesting. So what they have done is that using the northern blot analysis, they have characterized, so now this napoleon as well as the octopine, so they do have mRNA, that is because of the transcription. So now they have also characterized that these two has got put together, there are 8 mRNA species. They are very, very critical, in fact. Based on the northern hybridization assays, they have characterized that, I think, because of the transcript. So now each one has got different transcript. So now based on the characterization, they have given 1, 2, 3, 4, 5, 6A and 6B. Now this is in case of napoleon, whereas in case of an octopine, I think we have the 1, 2, 3, 4, 5, 6A, 6B, another one is going to be the 7. This one is not present in the napoleon, and whereas this 3 and this 3, they are not the same. So 3 and 7 is exclusively present, are different in octopine, whereas we do have a very good resemblance of all other 1, 2, 4, 5, 6A, 6B. It is highly similar between the napoleon and the octopine, whereas the transcript 3 and transcript 7 are different. Now, apart from these, we also have some virulent genes present in both of them. One is a TMS, another one is TMR, another one is going to be the TML. So now we have the TMS, TMR, TML. We know that because of the presence of this particular tDNA, the cells fail to differentiate into a plantlets or whatever the product which is required to be destined. So now, for example, they have done lot of studies of these individual transcript. Suppose like if you just remove, delete TMS gene, now then what happens? Immediately this particular cell or the tumor cell try to differentiate into a shoot. So now they know that because of the presence of this TMS gene, so the differentiation into shoot is blocked. Suppose if this TMR gene is eliminated or a knockout, so then this particular cell will be differentiated into a root. Suppose in this particular TML, so now this is going to be the size enlargement. So the cell is going to be enlarged. So in other words, the presence of these virulent genes, TMS, TMR, another one is going to be the TML, they are the virulent genes, so they suppresses the differentiation into either a shoot or into a root or the size enlargement. So they are very, very important in case of a tDNA. Okay, now this is the complete understanding of what exactly is a TI plasmid, and in the TI plasmid you know what is a tDNA, and on careful analysis the tDNA is again characterized into a napoleon type, another one is an octopine, and now this napoleon. So now apart from this transcript, so now the 3 transcript, it is napoleon synthase, NOS, and the 3 I told you they are different basically because now this is the octopine synthase. Now this is an enzyme, it provides napoleon synthase and this provides octopine synthase. We know how this enzyme synthesizes napoleon and another octopine. So here it is a pyruvate with arginine and here it is alpha ketoglutarate with arginine. So we are going to have the napoleon synthase and we are also going to have the octopine synthase. The most important one is going to be these 2 regions. So we call this as a border region. So now it has got basically on careful characterizations we have the 25 base repeats in fact.